So thank you, Mike, and thank you for having me here today. So I'll just get started. We all know that the gastrointestinal microbiota is a really important part of animal and human health. Paris has kind of alluded to this. It helps develop and educate the immune system. It's important for a lot of metabolic functions, and it can also provide pathogen exclusion or help with that animal excluding some potentially dangerous pathogens. I think our goal right now is understanding how we can actually use the microbiome as a tool to improve animal health, improve disease resilience, and of course, improve performance. So the three kind of areas I'm going to look at then are how does management, diet, and maternal phenotype kind of play a role in with the gut microbiota, as well as then health, resilience, and performance. So in the first kind of project, we looked at management and antibiotic use. So our objective here was to elucidate how antibiotics, in particular amoxicillin exposure, affects gut microbial profiles and innate immune function when piglets are challenged later in life with salmonella. So we actually just had two treatment groups, one that was fed antibiotics twice a day for 14 days, and then just a placebo group. Then after day 14, piglets were just reared normally, weaned at day 21, and reared in commercial setting until day 49. On day 49, we did an intraperitoneal injection of heat-killed salmonella, and then we looked at the immune kinetics at this time. So we looked at time point 0, 4, and 12 hours post this injection, and as well we have microbiota data to go with this. So what's really interesting is that although we have a 2-week antibiotic exposure period, this was only inducing a transient shift in the gut microbiota. So we actually see that cecal coliforms, so likely E. coli, was increased in postnatal day 7 and in day 14 in those piglets that received antibiotics. So they have this kind of coliform bloom. When we look at the sequence data, so this is actually live plated bacteria. When we look at the fecal microbial communities from sequence data, we see at postnatal day 3 kind of the separation between those animals receiving antibiotics and those receiving placebo. So these red and blue dots kind of separate, just showing that they're kind of distinct communities. But when we look at postnatal day 7, you no longer see the separation between these two different colors, meaning that the gut microbial composition is now quite similar between these two groups of animals. So these antibiotics are causing just really a transient shift. Things are kind of back to normal by day 7 already. I think what's really interesting about this, though, is that although the antibiotics only induce a transient shift in microbiota, we see that it actually has really long-term consequences on the innate immune system. So on day 49, when we did this IP injection, we saw that there was a rapid activation of leukocytes in the peritoneal cavity of these antibiotic-treated pigs. So just a little bit of evidence we see here at time point 4 hours, the antibiotic animals have a much higher ROS-producing cell number. And to go with that, actually at 4 hours as well, we see that NF-kappa B translocation is much higher at 4 hours in these antibiotic-treated animals, indicating they have a much kind of higher activation of these immune cells. To go with this, looking at peripheral blood circulation and RNA gene expression, we have kind of a Th1 type activation that looks like it's going on. So we have upregulated immune genes, including interferon gamma, TNF alpha, IL-6, and IL-2. So in the antibiotic-treated animals that are in red here, we see that interferon gamma gene expression is increased at 4 hours, which goes with that ROS-producing cell number, as well as that NF-kappa B translocation. And then with the TNF alpha and the 2 cytokines, we actually see an increased gene expression at the 12 hours. There seems to be an increased response of these antibiotic-treated pigs. One thing to note is we didn't go after 12 hours. So we don't know the kinetics after this time point. We don't know if this is a sustained response or if this is just an increased activation and this response will tail off. So the key findings of this is that neonatal piglet antibiotic exposure really does only cause a transient disruption in this gut microbiota, and we really just see this big bloom of E. coli. Likely this LPS has something to do with the stimulation or activation of the immune system, and this is causing some type of long-term innate immune consequence. We have rapid activation of immune cells and increased expression of peripheral cytokines. So what's next is we actually need to know whether or not this is a consequence or a benefit. I think that's still a question, what the actual kinetics are long-term and how this actually does affect disease susceptibility or resilience. So then secondly, we looked at how then diet and supplementation of piglets impacts their gut microbiota and gut health. And so then the objective of this was to determine the effect of supplementing a man-enriched fraction from a yeast. So we're using Actogen from Alltech on piglet gut microbial profiles as well as health. So this project was very simple as well. We just had 2 treatments, one of Actogen with 800 grams per metric ton, or a control. So it's obviously a very low inclusion rate. Piglets were reared just in a kind of a normal commercial setting and were crete fed from postnatal day 7 until day 21 on respective treatments. We had 10 litters of piglets. Then they were weaned into the nursery, kept on the respective treatments in 2 phases of diets, and then we just sampled at postnatal day 28. So 1 week post-weaning, we kind of think of this as like a crucial time, and not a lot of researchers are looking at this time point. And then we sampled later on at postnatal day 42 when they should have recovered from that post-weaning stress. So it's interesting with Actogen supplementation, we saw that there was an improvement in jejunal morphology on day 28. So I have Actogen in pink and the control group in orange, and we see here that the jejunal villus height is actually increased in this Actogen group, indicating they might have increased absorptive surface area, ability to absorb more nutrients and water, so potentially a better gut structure. But by postnatal day 42, we see that there's no longer an increase in this from Actogen piglets. Then when we look at the cecal microbial profiles, we see that Actogen does cluster separately from those control animals. So they have kind of a distinct microbial profile in comparison to those animals that weren't supplemented. And the bacteria that we're seeing that are changing seem to be a lot of mucosal associated bacteria. And one of them we see that is reduced in the Actogen supplemented animals is Campylobacter. So Campylobacter is something we've probably all heard about with human food poisoning, but it also does cause some piglet diarrhea. So it is an important foodborne illness that is important to the swine industry. So next, to kind of look at gut health with this, we looked at day 28 jejunal gene expression with an Affymatrix microarray. So we used the postnatal day 28, and if you see here, there's about 23,000 genes on a microarray chip. And when we put them all together, we see that there's a distinct clustering between the Actogen and the control animals, kind of indicating that there is differential gene expression between these 2 groups of animals. When we then plug in these genes into the IPA software, we can kind of get an idea of what these genes mean biologically. So we see that pathways are predicted to be upregulated in those pigs supplemented with Actogen, including the sirtuin signaling pathway, which is known to be an anti-inflammatory pathway, AMPK signaling, which helps to control barrier function in the gastrointestinal tract, some integrin-linked kinase, which is known to help with intestinal cell proliferation. We have the mTOR signaling pathway that's upregulated as well. We all know this is for protein deposition and growth, as well as STAT3, which is a known cell stress response. So there's kind of some indications and some biological functions that are linking to this change in gut microbiota. They have this increased villus height, so kind of indicating there is some improvement in gut health in these Actogen supplemented piglets. So key findings, that supplementing Actogen or maybe other feed additives as well can potentially reduce pathogenic bacteria. For instance, we found that Campylobacter was reduced, and this is something that Alltech and with Actogen has found previously in other animals. We also think that it may improve intestinal integrity as we see morphology changing, as well as all these gene pathways that are biologically indicative of that. So what's next? Doing a more robust performance trial, we had very low animal numbers, so the performance data here was not different, but the variation was quite large. And then kind of a natural challenge, are these animals actually resilient or resistant to disease? How does supplementing maternal animals versus the piglets, how does that work? And then we're also looking currently at other additives. So Actogen is just one, using a man-enriched fraction, kind of a fiber. There's other things such as acidifiers or enzymes that are, I think, commercially used already, but how these actually affect gut health and animal performance. So kind of the third part of what we're looking at in terms of host health and microbiome is how does the maternal host environment affect piglet gut health and performance? So sows consistently typically throw low or high birth weight litters, and we can phenotypically kind of group them based on that. And the implications of coming from a low or high birth weight litter are very long lasting. If you're a low birth weight, it's likely that you're going to continue to be a low weight animal, and you might have a lot of growth compensation to do before you kind of make up all of that less weight. So our objective is then to determine how sow phenotype, either low or high birth weight, influences the microbiome and gut health of piglets. So from this, for this project, out of 2,400 sows, 700 sows were selected, having over 3 parities recorded, and then we picked 20 sows that were either high birth weight or 20 sows that had low birth weight phenotypic litters, just as a sample set. From these 40 litters, we looked at the entire microbial profile of the piglets and the sows, and then we took a subset of these litters and looked at gut histology, microbial profiles, as well as the gut transcriptome, which I won't talk about. So just kind of the data on these litters that we did select, I think it's obvious between the high birth weight and low birth weight litters, there's quite a big discrepancy between the average birth weights, and the standard error is the same, and these are significantly different. I'm just going to draw your attention down here to the weaning weight, which I think is interesting. The high birth weight and the low birth weight at weaning, although there's about a kilo and a bit difference, there's a lot of more variation in this low birth weight litter set, and they no longer are significant. So although we've previously analyzed this as kind of based on their birth weight and the sow's phenotype birth weight, there might be something to be said about those pigs that do have compensatory gain and come out of the nursing, or when they're weaned, actually have the same body weight as a high birth weight animal. What does that actually mean?
This is litter birth weight.
Yes.
Is this piglet birth weight or litter birth weight?
This is
your high birth weight and your low birth weight. That's on a litter basis.
This is on a litter basis.
Litter basis.
Correct.
So the phenotype of high-low belongs to the sow.
Belongs to sow.
Sow,
Yeah. So
yes.
it could be that you've got variation because you've got high birth weight pigs and low birth weight pigs within your low birth weight.
Exactly. So right now we're going back into this data set to try and do exactly that. So this is where we're kind of at right now, just because we didn't see as much of a significant result as we thought we would. So thank you.
But John,
was wrong.
Yeah.
John, just your comment at birth weight, though, the variation's the same between the two groups.
Yes, variation.
Yeah, you're right.
Yeah.
We have to do some digging in this data set for sure. I think.
Variation's the same, but still that just means that you've got the range at birth. But I'm thinking the low birth weight individual from the low birth weight litter is a different pig than the high birth weight pig from the low birth weight litter. And I think maybe that's what's happening.
Yeah, or the impact is different. I think it's the small guys that are high the impact.
So we're currently working on that part of trying to reassess that so we can have a communication after that. So for jejunal morphology, we just see that there's kind of a negative correlation between crypt depth and birth weight, indicating that these low birth weight piglets tend to have deeper crypts, and this is kind of a known compensatory mechanism for some kind of assault on the villus. So that's kind of no surprise, but this is again with birth weight. And then when we look at the sow fecal microbiota composition, just to make sure that's not the variation we're seeing in these two groups of animals, they're basically identical. They don't really tease out at all between these two groups of sows. So their microbial composition is basically identical. What's really interesting is when we look at the piglet fecal microbiota composition, we do see that as a community structure, the compositions are similar, but when we tease out actual bacterial genera, we do see that Enterobacteriaceae is significantly higher in those low birth weight animals. So those low birth weight animals may, in fact, be more challenged based on their weight or based on their microbial kind of signature. So it's something that we're going to look into a little bit further.
Just remind me, what day is that collected? 28.
21?
They're at weaning. Yeah, 21.
21.
Yeah, they're weaning 21.
Yeah. So this is right at weaning. So the key findings here is that just sows with phenotypically high and low birth weight litters have similar microbial compositions. However, these low birth weight animals definitely have an increase in Enterobacteriaceae, so likely E. coli. What's next? We really need to regroup these animals and just see what the data says after that, and basically the importance of birth weight or weaning weight, or is it kind of their rate of gain during that period of time and how that all affects growth performance later on and disease resistance. So that's all I have today. Basically, I think there is a future potential to hopefully use microbiota as one of the tools to improve growth, health, and food safety. So thank you very much to